Okay, this lecture is going to focus on host microbe interactions and pathogenesis. Now, what you have to keep in mind is that there is some sort of interaction that occurs between the microbe and its prey, if you want to consider it like a predator-prey relationship. The prey being, of course, the host. When we talk about these type of relationships, the interactions influence virulence. In other words, the capability to cause disease, the capability to stay in the organism, etc. And we have to keep in mind at the same time that we all have microbiomes, in other words, microbiota. Okay, you've heard about the gut microbiome, lots of different bacteria. There's some that are on the skin, there's some in, for example, the vagina, there's some in the mouth. And what happens is that when there is some dysbiosis, which means a disruption of the normal microbiota, then we have problems sometimes. For example, antibiotics. A lot of women will say that if they're put on antibiotics, they end up having yeast infections. A lot of times what causes that, interestingly enough, is that the antibiotics will destroy the lactobacilli. These are bacteria that live normally in the vagina, and they create somewhat of an acidic environment by releasing lactic acid, okay, lactobacilli, okay? When they do this, the pH shifts in the vaginal environment such that it favors now the growth of yeast. Normally, the yeast organisms like Candida are suppressed because it's so acidic in that environment. And we'll just continue to explore some of these avenues as we talk about this. As you can see in the handout or in the book, we have the microbiota balance. In other words, for example, if you're doing well, you're not going to have any of these normal gut microbiota causing problems because think of it as sort of an ecological system. In other words, a bunch of different organisms, they interact with each other, everything works fine. Then you have a disruption. Person takes an antibiotic, for example. And so you start killing off some of the natural bacteria. The others will begin to take over. And then you start having this population explosion of certain types of bacteria, which a lot of times are detrimental to your health. In this case, we're going to talk about Clostridium difficile. And what happens there is they really begin to basically proliferate in the person. And then, of course, as the person basically will start having the illness of C. difficile, C. diff, as some people refer to it. And it's going to be on formites, which can be sheets, bedding, doorknobs, handrails, etc., as well as the healthcare workers' hands. Because there is a limited competition, because the natural ecological environment, I'm using that term, but basically the same as saying microbiota, is disrupted, the C. difficile will begin to take over. Nature abhors a vacuum. You may have heard that from time to time. And really what happens is that suddenly these particular types of bacteria flourish when someone has gone under extensive antibiotic treatment. And what happens then is diarrhea, pain, bloating, and basically the C. difficile will make things a lot more messy and a lot more difficult for the patient. Now, we're going to also mention about normal microbiota and opportunistic pathogens. Now, an opportunistic pathogen normally is not pathogenic because with a healthy immune system, you're able to keep it in balance, to suppress it, to fight it off. It's not going to cause any health problems. But in a weakened immune system, an immunocompromised patient, for example, everybody immediately thinks of HIV because the human immunodeficiency virus will destroy a lot of the immune function. But this can happen with a variety of other patients who are under chemotherapy, patients who have had tissue transplants, liver, heart, lung, etc. And so their immune system gets knocked out of kilter to prevent the rejection of the transplanted tissue. And that's when you have problems, because now the immune system's suppressed, could be by steroids, could be by a variety of immunosuppressive drugs. These other organisms will begin to proliferate and they will become pathogenic, causing disease. Now, tropism, remember that's a term. It's a preference of a pathogen for a specific host or a specific tissue of that host. When we're talking about COVID-19, first it was in bats, then it shifted into a host that could be human. In the beginning, we thought about it being just specific for the lungs, the respiratory system, but now we find that it is also present in the gut and can be shed in feces, etc. And these are things that happen. Remember, we talked about in influenza, antigenic shift, antigenic drift. Sometimes these mutations and sometimes these whole massive gene shifts will lead to sometimes a new preference for the pathogen to a specific host. It will shift to several other hosts, or it will actually increase its proliferation for various tissues in that host. So let's talk about virulence a little bit more. Excuse me. When we talk about host microbe interactions that influence virulence, we got to talk about what is virulence. This describes the degree or extent of disease that a pathogen causes. Now keep in mind that the term pathogenicity is the ability of the microbe to cause disease. So if you sit there and say, well, it's something that can cause disease, how profound is it? Is it the sniffles? It's a cold. If it's suddenly proliferating and destroying all of my lung tissue, well, that's something a little bit different. That has a nastier virulence. There are a variety of virulence factors which we're going to talk about, because these are means by which the pathogens can overcome our defenses. Now, when I talk about defenses, we're going to be talking about mentioning it briefly. We'll go into this in the next couple of chapters, but the innate immune system and the adaptive immune system. Okay? Now, most of you had AP2, Anatomy and Physiology 2, so you've already been introduced to this to a point. We're going to go into this a little bit later on and go into this much more in depth. But some of the pathogenicity factors are toxins, compounds that thwart the immune defenses, some that will acquire nutrients so that they can proliferate, others that will evade the immune defenses. You have to also keep in mind that not all pathogens are equally virulent, meaning that they can equally cause death, destruction, etc. No. Individual genetics, the mode of transmission of pathogens, the mutations of the pathogens, vectors, all of these will play a role in how virulent, how much this disease spreads. That's why when you've heard them talk about different types of influenza, there's some that spread much more rapidly, much more devastating. There's others that, I was tired for a few days. Remember also that the genetics of the host can play a role, okay? As I mentioned here, keep in mind that if the pathogen kills the host, speed and lethality will be balanced by time interval to transmit the pathogen to other hosts. In other words, what they usually find with a rapidly progressing disease, whoops, sorry about that, folks. Let's go back here to the very beginning, and I'll go into there. I just had a little accident there, and I apologize for it. And here we are. One of the things I want you to understand is this. Every once in a while, epidemiologists and infectious disease specialists will sit there and say, well, this is really virulent right now. And if the organism, which undergoes some mutations, doesn't kill the host, it will have enough time before it kills the host to spread to other hosts. Because let's face it, all organisms attempt to survive, to pass on offspring, to go to another host, in other words, okay? So what happens in some cases is a pathogen will basically begin to dampen down its effects. This is what we refer to as attenuated pathogens. They're weakened, usually by the loss of some virulent factors, to infect but not necessarily cause disease. And this may be used for vaccines for the pathogen itself. Now, it gets a little bit confusing. I hope it hasn't been too badly. If you have some difficulty, remember that you can always give me, and talk about this during discussion time online. But Basically, the point I'm saying is this. Think about it this way. If you want disease to spread through the whole population, you can't have it where the individual is sick, dies very, very quickly without having even time to pass the pathogens to the next population. Then you basically just, the pathogen wipes out a couple of patients and that's it. It's all done. You've limited the disease. But if a pathogen can spread for a while before it makes someone profoundly sick, it increases its chances that the pathogen will spread throughout the population, such as what we've seen with COVID-19. Okay, here are some of the review points here. Toxins. There is the lipopolysaccharide in gram-negative cell walls. That's a very nasty toxin. They will also, in some pathogens, have diverse secreted toxins, which we'll talk about in a bit. The AB toxin, the superantigens, things like that. Then we have also the immune system evasion, where they have either enzymes or toxins or the actual capsule itself, the structure of the pathogen that will help to allow it to evade the immune system. Some of them actually are just more transport mechanisms, but we'll get into that in a second. When we talk about adhesion, it's got to attach to something. If we're talking about the pathogen attaching to cells or to tissue, it's going to have various structures. The fimbriae, the binding effectors, some of them may actually bind to specific cell receptors on the host targeted tissue. That's why, for example, some viruses will only attach to certain cells in the respiratory system. If those receptors are not there, no infection occurs. Some of the other binding factors will also include what they call pili, which is a different type of extension from that pathogen. You see that a lot in bacteria, for example. Nutrient acquisition. Some of these pathogens will release enzymes to break down surrounding tissues to get the nutrients, break down proteins, amino acids, break down polysaccharides to monosaccharides, etc. The other thing that we're going to be talking about is iron binding proteins. Keep that in the back of your mind because it's interesting. Iron is absolutely essential for some pathogens to survive. The body may attempt to sequester and keep that from the pathogen, but the pathogen has other means to grab onto iron and hold onto it and bring it inside itself to successfully continue the invasion and the proliferation within the cells. Finally, we talk about invasion. There are various toxins, various enzymes that will break down and allow it to get inside, as well as flagella. Now, a couple of terms you do need to know. Cold, hard, and fast. Infectious dose 50. That's also known as ID 50. This is the number of cells, whether we're talking bacterial, fungal, parasitic, or viral particles, necessary to establish infection in 50% of the susceptible hosts. Some pathogens have a very low ID 50, meaning it's only a few cells once they get inside the susceptible host and they can start an infection. Others need a much greater number of cells to kind of jumpstart, overwhelm the initial immune responses, and then successfully, how can you say it, almost like landing on a beach to invade a new country, landing inside of a susceptible host to begin the infection. Now that's different, ID 50, from LD 50, lethal dose. A lethal dose 50 is the number of cells, again, bacterial, fungal, or parasitic. And by the way, keep in mind that when we talked about these different types of cells, it could be in the form of not merely living vegetative cells, but it could be spores, endospores, things like that, okay? Or the viral particles, as I mentioned before, necessary to kill 50% of the affected hosts not treated. You have to keep in mind, though, that these are rough estimates. They're mostly based on animal studies. You can't just line up 500 people and say, okay, everybody's going to get exposed to anthrax today. We're going to give you, some of you are going to get only a few cells, some of you are going to get a lot of cells, and we'll just find out who gets sick. Nah, that doesn't go well in society. So we use this as a rough estimate using animal studies, and it's helped to give you a score or an estimate of the hazards associated with the toxin or the pathogen. Now, if you take a look up here, this is table 10.1. Here's an example of difference. Now, keep them mindful of this, okay? Bacillus anthracis, the infectious agent that causes anthrax. In some cases, for cutaneous anthrax, in other words, you'd get this eschar, or it looks like a scar, or a blackened, almost like scab. You only need 10 to 50 bacterial spores, and you get that. That's cutaneous anthrax, but gastrointestinal, you need a much higher amount, in part because some of the means in which the spores are passing through the digestive system will be eliminated by things like the acidity of the stomach, some of the reactive enzymes, etc. Respiratory, though, there are some people that would debate that 10,000 to 20,000 number you see there. As a matter of fact, when I was at one bioterrorism conference, they said 1. Get it? 1. Inhaled into the lungs. That's it. You're infected. But that's basically stuff that's been known for serious strains of anthrax. Notice the LD50, 8.4, and you see that little symbol there? That's micrograms per kilogram. What we're talking about is millionths of a gram. Okay, but notice they used canines for the study. Now that's different from botulism toxin. Botulism toxin, the ID50 is not applicable. Okay, toxins, because they're not often made by infectious, because they are often made by infectious agents, are not themselves infectious, but they have devastating effects. And as you can see, 0.001 micrograms per kilogram body weight. Think about how absolutely small that is, okay? Either orally or injected would have a devastating effect. And these are, of course, based on non-human primates, okay? Now, we're going to get into the toxins. I want you to be clear about what toxins are. In this case, we can have endotoxins, which are mostly lipid-based. We can have exotoxins, which are released from the outside and sort of exocytotically released from the cells, and they're going to be present in gram-negative, gram-positive, whereas endotoxins are only gram-negative. The exotoxins are proteins. Usually exotoxins are coming from actively growing bacteria. Endotoxins, they basically leak out from the cell wall of gram-negative bacteria when the bacteria divide or die. We're going to get into some of the more details of this in a minute. Keep in mind that toxins, it's a very broad definition. Molecules that in small amounts generate a range of adverse host effects. This can include tissue damage. It can involve suppressed immune responses. It can lead to tissue death. When we say something is toxigenic, this is a pathogen that generates a toxin. When we talk about the term toxemia, that's when the toxin spreads from local to systemic. So it's now spread by the bloodstream. It may then cause serious damage to a variety of tissues and organs. For example, anything that does a lot of filtering, liver, kidney, that's when you're going to see toxemia have some really quick, clear signs, because these organs are involved with filtration, going back to your anatomy and physiology too, and you can see how this will cause a lot of destruction. You'll end up with multi-organ failure. First organs probably to fail will include the liver and the kidneys. Now, some organisms make a toxin and may not survive to benefit from the toxin's effects. For example, Bacillus cereus is involved with food poisoning. It has a heat-stable toxin. So even if we cook something and Bacillus cereus was there making the food go bad, the Bacillus cereus may be destroyed, but that heat-stable toxin is still there, making the patient sick. Toxins are defined as having defined in two major classes: endotoxins, exotoxins. Endotoxins, as I mentioned to you before, are produced by gram-negative bacteria, usually from the outer membrane. They're released from the killed gram-negative bacteria cells. That's supposed to be gram, sorry about the spelling error. One very clear example is a lipopolysaccharide, LPS. It's the lipid portion called lipid A, is the endotoxin. As a matter of fact, this is so nasty that you really, really have to make sure things like intravenous solutions, etc., are tested well beforehand for even the most, we'll say, microgram amounts. That's where you get into using horseshoe crab blood. There's a material called LAL that is extracted from the blood of horseshoe crabs that interacts with even a billionth of a gram of lipid A. And if you have that, then you know the product cannot be put into the intravenous solutions, things like that. Endotoxemia is endotoxins are entering the blood, and the symptoms include fever, chills, body aches, hypotension, tachycardia, increased respiratory rate, inflammation, nausea, and vomiting. In other words, everything's going crazy. Now, if you have this in high doses, this can lead to septic shock, otherwise known as endotoxic shock, and death, usually by organ failure. There is something I wanted to bring up that's not mentioned in the book, but it's very important. It's called the JARISCH-HERXHEIMER reaction. Now, this is a reaction to endotoxin-like products released by the death of harmful microorganisms within the body during antibiotic treatment. Main point. You will see a JH reaction. A lot of times when a patient is on antibiotics, they start feeling better, but then they start having these weird symptoms. And they'll start saying, I'm having inflammation, I'm having chills, etc. And what it is that the antibiotics are killing the cells of the bacteria, but the products are being released into the systemic circulation and causing an inflammatory response. This is normally seen with antibiotic treatment of syphilis, but with a lot of other spirochetes, you'll see this also. The spirochete bacteria include things like, Lyme disease, relapsing fever, leptospiroidosis. The interesting thing now is that since Jarisch and Herxheimer, roughly around the turn of the 1900s, had described the reactions. We've seen this with Q fever, Bartonellosis, brucellosis, trichinellosis, and African trypanosomiasis. And guess what? You can even see it in certain cases with treatment for E. coli. And you're kind of like, but wait a minute, isn't that necessarily? Yeah, it's that's gram-positive. Mostly what you have is prophylaxis, treatment with an anti-inflammatory agent. Now get this, NSAIDs, non-steroidal anti-inflammatory drugs. Aspirin, yep. Ibuprofen, yep. But not steroid drugs. Only non-steroidal anti-inflammatories will work. Okay? All right. Here you have the LPS, here you have the endotoxin, and you can see lipid A is basically extended out from the cell. When the cell dies, it releases this, and the endotoxin is released as the cell begins to break up. Now let's talk about exotoxins for a few minutes. These are toxic soluble proteins. They're released by both gram-positive and gram-negative cells. Exotoxins are named for either the pathogen producing them or targeted tissue organ that the toxin lacked on. We're going to go through several different types. Hint, hint, yes, you need to know these. Type 1 is a membrane-acting extracellular toxin, otherwise called surface-acting toxin. It tends to alter the host cell physiology, and it could be anywhere from changing the cell physiology, the biochemistry going on in the cell, to actually affecting gene expression. Now, there's a class that's part of type 1 called superantigens. They act a little bit different, but a little bit very similar. They overstimulate the host immune system, causing massive inflammation. They can cause fever. They are part of what they refer to as pyrogens. Superantigens are the ones that are going to cause sometimes massive reactions for certain patients. What about type 2, type 3? Type 2 is a membrane-damaging toxin, and it forms pores in the membrane. Or it may actually remove the phosphate groups on phospholipids. Basically, it acts like an enzyme and pops off the phosphate. And so what you end up having is the destruction of the cell membrane, which leads to eventually the cell lysis. Type 3, though, is an intracellular toxin. It enters the cell to exert its effect. This is where you get into what's called the AB toxins. It has two parts, okay? What happens is this. Part A is part B is the binding part. Sorry about the typos. And B allows entry into the cell, sort of attaches on. It's then part A that gets broken off, that exerts damage inside of the cell. And you'll see these different ones listed here. Don't worry, I'm not asking you to memorize each and every one of those, but get familiarized with them, because 1, 2, and 3 are important, and you will see them in your allied health, nursing, pre-med type of subsequent coursework, okay? And you can see this. Here's type 1. Toxin comes in, attaches to a receptor, begins acting on the inside of the cell. Type 2, you begin starting to form these pores, and what happens is that you literally have the phospholipids, for example, starting to break down. So you go through this hydrolysis and popping off phosphate groups, etc. So you literally have a breaking down of the membrane lipids. Type 3 is AB, all right? You have one section, B, binds the plasma membrane, successfully delivers it on the inside. Then A and B separate. A starts doing the massive bad stuff inside of the cell. Now, when we move forward, we're going to talk about the 5 steps to infection. Number 1, a pathogen must enter the host. Number 2, the pathogen must adhere to the host tissues. 3, the pathogen must invade tissues and obtain nutrients. 4, the pathogen must evade the host immune defenses so that it can replicate. And then the 5, and this is where it's also important too, is the pathogen must be transmitted to a new host to repeat the cycle. Are all these steps important? Absolutely. Let me just give you a comparison. Think about this for COVID-19. One of the things that they found recently is they found evidence of COVID-19 genetic material in sewage. Now you sit there and say, ugh, sewage, what does that matter? Now we know that COVID-19 can be brought in by either the oral to the basically into the gastrointestinal area, because there are ACE2 receptors there. It can also be brought in by the respiratory system. ACE2 receptors there too. Causes a lot of havoc in the respiratory system. Everybody talks about respirators, but what you don't remember them talking about is if it's in the GI tract, it has to be shed out, and it's shed out in fecal matter. Fecal matter that can also be detected when you have microbiologists, infectious disease specialists, checking sewage treatment plants, and they'll find out, yep, we got COVID here. So if we've got COVID here, we've got strong evidence that it's passed and shed out. Something has to, once it goes in, has to come out to be transmitted for a new replication cycle in a new host, okay? So let's explore these a little bit more here for a minute. The pathogen must enter the host. Now there are a variety of portals of entry, which you'll see in a diagram in a minute. The portal of entry are basically sites where the pathogen uses to enter the host. It could be arthropod-borne, it could be through sexual contact, it could be fecal-oral, and that's where you get into COVID-19 being fecal-oral. But also the thing is that, yes, it can include respiratory tract. It can be aerosolized, such as in someone sneezing, coughing. But not only is it merely the respiratory tract, there's evidence that COVID-19 can be ocular. So when we include a variety of other sites for portals of entry, you have to keep in mind skin, ocular, otic, optic, which is basically through the ear, parenteral, okay? In parenteral, you're bypassing the skin. Entry is via hypodermic, through the hypodermis or the muscle. Could be cuts, abrasions, punctures, injections, things like that, okay? Respiratory tract, it's pretty clear what happens. You breathe it in, it gets into either the trachea or into the lung tissue, and there you've got it reproducing and causing damage. Gastrointestinal tract, the GI tract, the urogenital tract, as well as transplacental entry. When we talk about transplacental, it's crossing the placenta to infect the fetus. Keep in mind also that a lot of these other ones we've talked about are mucous membranes. The gastrointestinal tract, basically in the mouth. You're talking about also in the respiratory tract, etc. Now, here is a variety of these. I would like you to get familiarized with them now. That's why we try to tell people things like, okay, don't share needles. Hepatitis B and C for the parenteral. Keep in mind, for example, we talked about the host biome. Hey, there are staph germs that are on your skin. They're fine. They're kept in control. Cut the skin, such as a knife wound or an accidental cut, or worse, a gunshot wound or anything else. And now they're being put inside where it's warm, dark, moist, lots of nutrients, etc. Ocular, simple. Conjunctivitis, various bacteria, pink eye, and other viruses can get in there. And we can go on and on and on. So take a look at all of these and take some time to review them, okay? So let's move to step 2. Pathogen must adhere to host tissues. There are a class of virulence factors called adhesins. Basically, they allow things like viruses, bacterial, fungal, protozoans to basically use these proteins to attach to a host cell in a specific or non-specific manner. Specific is sort of, well, okay, it favors, let's say, the respiratory cells in the respiratory system, basically the epithelial cells in the respiratory system versus, let's say, the digestive tract, something like that, just to give you a concept there. Non-specific would be attaching to anything. Now, adhesion factors can be useful for vaccine development targets. An example of that would be there are certain vaccines against pneumonia that have been used with the the lipopolysaccharide of a particular type of bacteria, for example. Now, the other issue you have to be mindful of when we talk about step 2 is biofilms and quorum sensing. Remember, quorum sensing is that basically it's communication means where other bacteria can tell each other, okay, there's enough of us here, so we can now activate our genes to make biofilms to enhance our survival, our protection against antibiotics, our protection against white blood cells, etc. And that enhances our infective capability. So they make the biofilm, which is basically a lot of polysaccharides, some proteins, etc. And they thrive there, okay? Now, here are some examples of pathogen adhesins, fimbriae and pili, silastic acid binding factors, heparin, heparan, sulfate binding factors, and fibronectin. And you can see the various types of bacteria. Don't worry, I'm not asking you to memorize each and every one of those bacteria types or viruses, etc. But get the idea that it's something that's there that allows it to adhere to and begin its process of getting ready to go to step 3, which would be to go inside the cells. Here's a couple points about biofilms. Biofilms exist on urinary catheters, hip or knee implants, the subvenous catheter, as well as dental plaque, and it's believed to be also playing a role in kidney stone formation. Okay, now we're at step 3. Pathogen must invade the tissues and obtain the nutrients. In other words, invade, get nutrients. Why nutrients? Because you're going to need them to survive, A, and B, to proliferate this pathogen and then be able to replicate and spread to other parts, other cells, and eventually shed off to infect other hosts. The main point is that invasion leads to host cell damage. You have factors called invasins. They're local acting factors that allow pathogens to invade host tissues. They may be membrane-assisted or secreted enzymes. How do we obtain nutrients? Well, one of the best examples is iron. Siderophores are iron-binding complexes. They're used by bacteria to sequester iron. Now, there are some extracellular enzymes that are secreted by bacteria, fungi. That's how they also do it. If you give an example really quickly, fungi will secrete certain enzymes out of the hyphae, the extensions on the fungal cells. And what they do there is they basically start to break down the surrounding environment. Proteins are broken down to amino acids. That's where you get things like athlete's foot and things like that. They're basically breaking down the surrounding keratin tissue to take in the amino acids necessary to continue living. Now, there are also some cytopathic effects. These are effects caused by pathogen invasion and damage to cellular processes. We're going to get into more of that later. Here are a couple of invasion examples: flagella, collagenases. Remember, that's ASE. That's basically telling you, ta-da, it's an enzyme and it breaks down collagen. You'll see this with Clostridium for perfringens. It doesn't sound familiar, but it's also known something a little bit easier to remember: gas gangrene. Yes, this is the bacteria that breaks down tissue and will actually form some types of gas as it's creating this infected tissue and dead tissue, okay? Neuraminases, coagulases, and kinases. These are also doing the same thing: invade, destroy tissue, take up the nutrients, okay? Here is once the infection occurs, what happens next? Now, this is a perfect example here. It looks like Shigella bacteria. What you would have is they remain on the surface, or they may actually reside inside the cell, but they don't stay there. They may actually pass between the cells, as you can see in some cases, but in other cases, those basically what look like cilia or flagella will help the cell propel itself into deeper tissue, passing from one cell membrane to the next to the deeper tissue, okay? Finally, we have 4. Pathogen must evade host immune defenses so it can replicate. You have to understand this. The bacteria or other organisms, for example, some protozoa, what they have to do is they have to create sort of a smoke screen, some sort of protection against the immune system, the white blood cells, antibodies, etc. So some of this might be by use of biofilms. Others are by intracellular defenses. They may actually get captured, phagocytized, but then be able to escape from the macrophage. Or they may actually destroy the macrophage in the process from the inside out. Then there is latency. There's also other characteristics we call antigenic masking, mimicry, and variation. What some, for example, Leishmania and several parasitic worms will do is they will change the outside antigens of their exterior to avoid the immune actions. Sometimes what they'll do is they'll change it, switch it up day in, day out, or every few days or every few weeks or whatever. So even if the immune system starts to build up an attack pattern looking for a particular antigen on the surface of the organism, the pathogen, by the time that you really get a full-blown immune reaction, too late. The pathogen has switched its surface antigens, and now the immune system can't find it. And the pathogen continues to proliferate inside the host. Now, there are other ways to undermine host immune responses. Interference of phagocytes by including release phagocyte-killing toxins. Avoid the phagocytosis with a capsule. So basically, the capsule prevents it from either being brought inside or broken down once it's inside. It'll block the phagosome fusion with the lysosome. Remember, the lysosome is that small vacuole with digestive enzymes. It may also escape the phagosome once it's brought inside, and then it will end up living inside the phagocytotic cell. An example of that is tuberculosis, okay, bacteria. or it may actually neutralize the hydrolytic enzymes of the lysosome. When the phagosome and the lysosome fuse, it forms the phagolysosome. Okay, if you remember that. And then we also have immune suppression. It will inhibit the immune responses or destroy the immune cells or the cytokine release. Now, you might think about that from HIV, but there are a variety of other pathogens that do this to a lesser extent, and they survive as a result. Here are some of the strategies that will happen. The mechanisms for escaping host immune defenses include, as we've said, let's hide. Latency, intracellular lifestyle, antigen masking, mimicry, variation. I got a little cartoon of that coming up. The undermine basically will be to suppress the immune function and to avoid phagocytosis, or after you've been brought inside, prevent the phagolysosome fusion, or even if you're exposed to the digestive enzymes from the lysosome, basically you can neutralize them. Here's that little cartoon here, the antigen masking. So the pathogen covers itself in host factors to avoid immune detection. Antigen mimicry, it basically resembles the host molecules, helping it to evade immune detection. And finally, variation. Continues to keep switching them. Leishmania is one of those, and there are a variety of other ones, including, I believe, the malaria parasite that does that too. Finally, we've got messing up phagocytosis. As you can see in this diagram, here we have it. Okay, so basically the pathogen gets consumed, taken in by the phagosome, the lysosome fuses, and it's supposed to die inside the phagolysosome by the digestive enzymes, destroying the pathogen, But that doesn't always happen. Pathogens will release toxins, and they will kill the phagocytes. Pathogens will have an outer capsule that will avoid the phagocytosis. Pathogens may also basically block the fusion of the phagolysosome, or will break out of the phagosome itself. And in some cases, it'll just neutralize the hydrolytic enzymes delivered by the lysosome. Finally, step 5. The pathogen must be transmitted to new hosts to repeat the cycle. Now, we've talked about portals of entry. We're going to talk about portals of exit. Route of a pathogen that must take to exit the body, particularly to infect other hosts. And by the way, I've got something that I put up on one of the weeks that is the chain of infection, includes the different types of portals of entry. Print it up, review it. The disease reservoir. That is a term used for any environment or organism in which the pathogen tends to naturally thrive. This can be biotic, in other words, living. Bats, humans, prairie dogs, raccoons. What do you mean, bats? Well, that's what they think COVID-19 came. Humans. Well, yeah, if you have one person who is infected with malaria and the mosquitoes basically get blood from them, they can then allow the mosquitoes to then transmit to other people infected. Or we could be talking about the common cold. Prairie dogs. Well, prairie dogs can have fleas on them. And these prairie dogs can have plague bacteria that are in the fleas. Raccoons. Well, have you ever heard of raccoons being rabid? Yes, rabies. This can also be water or soil. One quick example. Over a couple of hundred years, we had cattle drives, particularly in basically, let's say from 1800 to 1900, 1950, whatever. And in those cattle drives, the soil has the presence of Bacillus anthracis. Anthrax, yeah. So anyways, what happened was, when one of the presidential conventions occurred, there was a bioterrorism monitoring unit called BioWatch that was put nearby the convention to see if, just to basically warn people if there was an attack. Turns out the BioWatch device went off and tested positive for Bacillus anthracis. And as I remember the head of it telling me this very succinctly, he said, we didn't realize it, but what happened is this. A lot of the soil in there had anthrax bacteria. What stirred up the soil, that eventually the soil moved in and activated the BioWatch unit for anthrax, was the generator that was connected to the CNN media truck. The generator was providing power, and it was also stirring up the soil. True story. Also can be formites, inanimate objects, doorknobs, handrails, hospital room surfaces, all these different types of things can be part of the cycle so that the disease is passed on to someone else. Okay? And don't forget aerosols. Okay? When somebody coughs, sneezes, the particles might be very minuscule, but they may contain the virus or bacteria. Okay, and you can see all of these as being out here where you can have these secretions occurring, okay? Now let's talk about safety and healthcare, because this is where we get into stuff that's interesting. You have to think about this. Biosafety levels dictate appropriate on-the-job behaviors in healthcare. And it's important to know the biological hazards and the risks. So this enhances the worker's safety and helps them to protect themselves for pathogens. This is where we get into what we call BSL, biosafety level. These are safety protocol levels based on the following criteria. 1, level of infectivity. Not necessarily the level of contagiousness, but can this be infectious, period. Extent of disease caused and the mortality rates. People aren't going to worry as much with a cold virus, but they will be with COVID-19. Or better yet, Nipah virus, which has a, 40%, I think it is, mortality rate. Mode of transmission. Is it transmitted by air, water? Is it transmitted by contact? Anthrax is not transmitted from person to person. But if a person gets exposed to the endospores, they will be infected. And are there availability of preventions and treatment of the disease? One of the problems with some pathogens is that we have no means to treat it, just basically palliative care, make them feel comfortable. That's why everybody's going bananas over hydrochloroquine and zinc sulfate as one means to possibly treat, but some people have objections to that. The other issue is, do we have a vaccine? Well, we've got vaccines against rabies, and we've got vaccines against, let's say, influenza, but we don't have vaccines that are really effective against malaria. So there'll be a higher risk to the people doing the work. Now, this, of course, means that it allows, by understanding the BSL levels, workers to properly to use proper safety practices, protective gear, and facility features to minimize exposure risks. And this is where I want you to review the chart and understand the levels. Here's the two individuals here, and you can see these are pressurized suits, okay? Even the headgear itself is sort of a bubble. Here is basically a biosafety hood. But this air connected by this coil here is connected in such a way that an organism could not even get inside the suit if there was a small leak. If there was a small leak, it would be air evacuating out, but not allowing it to get inside the person's suit. If you notice, that suit is pretty much integral across the entire person. So, even the booties and the wrists, and they're probably double gloved also. So this is probably a BS level. My guess, and there's also a shower in the back, I'm going to basically take a guesstimate and say it's a BS level 4. Okay? Now, here's the thing. We've worked in labs where you have BS level 1, just hand washing, work can be done on an open bench. And of course, basic safety protocols demand that no food, beverages, or even chewing gum in the lab occurs. You don't need any PPE. BS level 2, though, you do need lab coats and gloves. You may need a safety glasses or face shield if there's a splash risk. BS level 2 plus is when you're starting to mess around with really slightly infectious more agents like Salmonella typhi, rabies virus, Zika virus, HIV, in some cases, hepatitis C, dengue virus. But then you get into BSL-3. Now look at some of the organisms here. These would be really nasty ones. Therefore, the agents are manipulated in a biological safety cabinet. Controlled access and authorized personnel, they do background checks on the persons, okay? People entering the area are warned of risks. They're vaccinated if possible. They're monitored for infection. They decontaminate all the waste, decontaminate the labware, special airflow management, self-closing, and double door access. Okay? Then you get to BS level 4, and that's where you get into the space suits. Full body hazardous material suit, air piped into the suit through specialized air delivery. Now, you have to have appreciation for all of this. This is to minimize any transmission, not just to the workers inside, but having it leak out beyond the lab. Okay? What's one of the things that's been talked about recently with COVID-19? That the Chinese lab that was about less than 3 miles away from the open live food market may have been working with viruses and had only a minimal BS level 2. In other words, some lab coats, some gloves, but definitely not at the level of 4 or 3. And they may have actually had accidents where they had leakage of material outside. Okay? So let's move forward. Now we talk about universal and standard precautions. Okay, what's what? There was a change in mind when we got into about the 1980s. We went into universal precautions. These were guides set up to prevent blood-borne pathogen transfer. In other words, when you were working with blood or bodily fluids, the big concern, particularly in the 80s, was HIV, to a lesser extent, Hep B, Hep C, but Hep C wasn't called Hep C yet. It was called non-A, non-B, hepatitis. This changed. Now, later on, when you start getting into about nearly the late 80s, early 90s, the Center for Disease Control expanded the policies. They basically said, look, regardless of the infection status of the patient, regardless of what you're working with, all patients should be treated as potential sources of blood-borne or infectious agents. So they moved up the level. They no longer called it universal precautions, it's called standard precautions. Sometimes people will use the universal precautions as an older term, but they will still hold up to the higher standards. The standards are basically now all the precautions exist for handling any bodily fluids, including blood, urine, feces, sputum, vomitus. You don't want it in contact with membranes, non-intact skin, or fresh tissue. And this means a lot because you have individuals who, for example, with herpes virus, I remember one case of a doctor who was an obstetrics doctor, and she had double surgical gowns, gloves, but there was a space at her wrist. And when the fluid, when she was delivering the baby of the mother who happened to have herpes, she got a small splash of the fluid onto her wrist, and that's where she was infected with, herpes. So a lot of this has changed. We don't necessarily sit there and say, look, have you had sex with this, or have you been intravenous? We don't even mess around and ask the patient. We presume that there is the potential for that. That doesn't mean we accuse anyone of something, but just that we take the safety protocols very, very seriously. These include, of course, when you have new gloves. They're changed for each new patient and for each new procedure. So you keep changing gloves. That's why what happened in the late 80s was there was a sudden drop in the available supplies of gloves. You had companies that were going completely 724 making latex gloves, particularly in the South Asia market. They were going like gangbusters because there was a shortage of gloves. And this is particularly because of the concerns with HIV. They, of course, moved up to a new type of glove. You also had different types of gloves available. We also went into this, basically, it's almost now, the best way to put it is, it's almost like breathing for doctors and for medical personnel. You go in, you wash your hands before you see the patient. After you're done, you wash your hands again. And that reduces the chance of picking up anything or transmitting it from one patient to the other. Barrier clothing and face shields or masks if there is a splash risk. Now, in China, when they were dealing with COVID-19, you would see them having eye goggles or a face shield. Why? Because when somebody would cough inadvertently, they wouldn't be hit with any of these small particles, because we started to see evidence that maybe the infection was not merely through the respiratory system, but also through the eye. Also, properly managed BioSharp buckets. BioSharp buckets were atrocious. I know this when I worked in surgery years and years ago, that basically they would fill the buckets till they were overflowing, and you could get puncture possibilities. And that's where some individuals actually got diseases like hepatitis B or HIV. The surfaces were disinfected before and then maybe necessarily afterwards, and everything else. Contaminated laundry or garments are removed and laundered as soon as possible. So here's where you see a lot of the basics now. Standard precautions, hand hygiene, gloves, basically protection if there's a risk of exposure to wounds, bodily fluids, mucous membranes, barrier gowns and face shields when splash risks exist, and disinfection. And you can see and review all of this. Of course, this has become sort of second theme now, because in part of COVID-19, everybody's been brought back up to speed on this. Now, here, of course, is an N95 mask. Everybody's trying to make these as quickly as possible in the midst of the situation. Now, what an N95, you have to understand clearly, is 95% of the very small particles are blocked. That does not mean 100%. If you want a better mask, they do have N100s. They're more expensive, but 3M has perfected the techniques, as well as others, to basically have masks that will prevent the microparticles from getting in. Here's a brief refresher here of the safety precautions as well as the biosafety levels. Minimal risk at BSL1, BSL4 is the highest risk. Remember that in your labs, when you were doing those experiments, the most you had a BSL2 risk. Now, as we finish up on transmission precautions, they're taken as well as standard precautions. When transmission precautions are in place, signage must notify others, in other words, signs here and there and everywhere else, of the visitors and healthcare personnel that these precautions are in place. And a lot of these seem just sort of like, well, it makes sense. Contact precautions, minimize transmission of infectious agents spread by fomites and to healthcare workers' hands. Gloves and gowns. One of the interesting things was a study that came out about a year or two ago where they said that basically, if you know those panels that we push doors open with, those metal, if they were made of copper, the interesting thing is pathogens don't last long on them. And so they reduce or minimize the risk of transmission. Droplet precautions prevent the spread of pathogens in cough, sneeze, droplets. Masks where possible. The patient has, if you can do it, a private room. Now, in this day and age of this pandemic of COVID-19, it makes it quite difficult. So we put them into isolation, even if it means that you have several patients in the same room that have the same disease. It's not the best, but right now we're facing a pandemic. Airborne precautions to counter the fine respiratory aerosols. You may have things like HEPA filters. You may have an airborne infection isolation room, AIIR. This is where you see what's called negative pressure rooms. Now, what negative pressure rooms are is literally the air set up so that it stays in the room, goes through a venting system, but it doesn't release air back into the room. Actually, if you open the door and you put like a feather or something nearby the edge of the door, you'll see that the feather is pulled into the room, not leaving the room. The main purpose is that the airflow will always stay in the room. So air might come from the outer hallway to the room, but it will not go from the room to the hallway where it could infect others in the hallway. I hope that makes it clear. Okay, as we finish up, I would encourage you to review the summary on page 319, the chapter overview on page 320. Go over the end of the chapter questions on 322, 323. Remember to do your homework and the quiz in Mastering Microbiology. Review the videos in Blackboard for lecture content. The next section we're going to go into is innate immunity. Have a wonderful day.